Hello everyone and welcome to today's session. And today we'll be discussing the NCERT exercises for the chapter Body Fluid and Circulation. This is the disclaimer. So what are the objectives of today's session? So today we'll be discussing the basic concepts related to the chapter Body Fluids and Circulation. So we'll be discussing the NCERT exercise questions. These questions are based on some concepts. We'll discuss these concepts by working through NCERT exercises. Okay, so in this session, there'll be 14 questions. Clear? And these questions are related to the chapter Body Fluid and Circulation. Okay, so let's begin with the first question here. So first question is, you have to name the components of the formed elements in blood and mention one major function of each one of them. So let's have a look at the answer for this question. Okay, now the components of blood are as follows. So first one is the erythrocytes. Now they are the most abundant cell and contain the red pigment called the hemoglobin. So erythrocytes, they are also known as RBCs or red blood cells. Now hemoglobin, this is rich. This is an iron-rich protein that gives blood its red color and is responsible for transporting oxygen from the lungs to body tissues and carbon dioxide from the tissues back to the lungs. So these RBCs, they consist of this pigment hemoglobin. Now these cells, they carry oxygen to all parts of the body. Now red blood cells, they are continuously produced in the red bone marrow, which is located in the spongy tissue of certain bones like the vertebrae, ribs, breastbone, as well as the pelvis. Now, production of these RBCs, this is known as erythropoiesis. Now, let's talk about the range of the RBCs. So, range is about 4 to 6 million. Clear? So, 4 to 6 million RBCs per cubic millimeter of blood is present. Okay, which typically ranges being, so the range in females and males are, so in females it is about 4.5 to 6 million and in males it is 4.3 to 5.9 millions. Okay, clear? So this is the range of the RBCs present per cubic millimeter of blood. Okay, now let's continue with the solution. Let's talk about the leukocytes now. So these are basically white blood cells and they're crucial for the immune system. So leukocytes, they are the colorless cells. So unlike red blood cells, they don't have hemoglobin. That's why they are not involved in oxygen transport. Now you can see these cells, they do not consist of the hemoglobin. So this is a key difference between WBCs and the RBCs. Now leukocytes are the largest cells in the blood. They are much bigger than the red blood cells as well as the platelets. Also, they can be divided into two main categories, which we are going to discuss now. The first category is that of the granulocytes. So these are the white blood cells that have visible granules in their cytoplasm. Okay, so granulocytes includes neutrophil, eosinophils, and basophils. The granules in the cytoplasm contain chemicals that help them fight infection and respond to allergies or inflammation. Now, neutrophil and phagocytosis cells, they protect the body against various infecting agents. Clear? So neutrophils are like phagocytosis cells. Eosinophils, they are mainly associated with allergic reactions. They help control the body's response to allergens. Basophils on the other hand, are involved in inflammatory responses, releasing chemicals that help start the healing process. Okay, now let's continue with the discussion. Next one is the agranulocytes. So these are type of white blood cells that lack visible granules in their cytoplasm. Agranulocytes include lymphocytes and monocytes. Lymphocytes are responsible for immune response, while monocytes help by engulfing and digesting the foreign particles. So lymphocytes act like a body's defense strategies, okay, organizing attacks against invaders, while monocytes are like the cleanup crew, removing harmful substances through phagocytosis. Now let's discuss about the platelets. So these platelets, they are not true cells, but they are small fragments that play a crucial role in blood clotting. Platelets are small, irregularly shaped bodies found in the blood. Okay, these platelets contain chemicals that are essential for the clotting process. When the vessel is damaged, these chemicals help form a clot to stop bleeding. Main functions of platelet is to promote clotting. Without them, even minor injuries could lead to dangerous blood loss. Next question, what is the importance of plasma proteins? So let's have a look at the answer. Okay, so plasma is a colorless fluid of blood that helps transport food, carbon dioxide, waste product as well as salts. Plasma makes up to 55% of our blood. This means that more than half of what we call blood is actually this plasma. Okay, now about 6.8% of plasma is made up of proteins like fibrinogens, globulins, and albumins. Each of these proteins has a specific job, much like different employees in the company. Now, fibrinogen, this is a plasma glycoprotein made by the liver. It's like a special ingredient added to plasma to help blood clot when you get injured. Fibrinogen main job is to help blood clot. So when you get a cut, fibrinogen helps form a mesh or a net that stops the bleeding. Okay, so let's continue with the answer in the next slide. Clear? Let's talk about the globin. Globin protein. This is a major protein of the plasma and they're very important immune functions. Globin's role is to protect the body against infecting agents. Okay, now let's talk about the albumin. This is another major plasma protein. Albumin is the most abundant plasma protein and has several key roles. Albumin helps maintain the fluid volume within the vascular space. It's like a sponge that keeps water inside the blood vessels, preventing swelling in the tissues. Okay, now this is the column-based questionnaire. Here, two columns are given to you. Okay, in the first column, you can see what is mentioned: eosinophils, RBC, AB group, platelets, and systole. You have to match the column 1 with column 2. Clear? So let's have a look at the answer. Okay, so you can see eosinophils are white blood cells that help resist infections. So A goes with 3. Red blood cells are responsible for gas transport in the body. Okay, that's why B matches with 5. Then the AB blood group makes an individual a universal recipient because their blood lacks the antibodies against both A and B antigens. Okay, so C goes with 2. Then platelets are crucial for blood coagulation. So D matches with the 1. And then systole is the phase of the cardiac cycle marked by the contraction of the heart. So E should be, or E is matching with the fourth. Okay, so this is the correct answer. Next question, why do we consider blood as a connective tissue? So let's have a look at the answer. Okay, now let's start by understanding the basic property of the connective tissues. So connective tissue have cells that are not tightly packed, but instead are scattered throughout an extracellular matrix. Now, this matrix acts like a background material similar to how jelly holds fruit pieces in a fruit jelly desert. Another important feature is that connective tissues serve to connect different body systems. Next one. Now let's focus on the blood. Blood is considered a type of connective tissue for two main reasons. Okay, let's discuss these reasons. First, blood is mesodermal in origin, just like other connective tissues. The mesoderm is the middle layer in the developing embryo. So this is one of the germ layer, and it gives rise to tissues like bone, cartilage, and blood. Now, this shared origin is a key reason for blood's classification. Second, blood connects the body systems by transporting oxygen and nutrients everywhere and removing waste products. Okay, this is clear. This is another function of blood. Finally, blood has an extracellular matrix called plasma. Within this plasma, red blood cells, white blood cells, and platelets float freely. Okay, clear? This structure is typical of the connective tissue. Moving on to the next question. So here the question is, what is the difference between lymph and blood? Okay, so let's have a look at the answer. Okay, this is a table showing you difference between lymph and blood. So lymph is a colorless fluid that does not contain red blood cells. Lymph has higher concentration of white blood cells compared to blood and is primarily involved in the body's immune system by filtering out the pathogens. While the blood is red due to the presence of RBCs and is responsible for transporting oxygen and carbon dioxide throughout the body. Okay, so lymph, you can see this is a colorless fluid, and then blood, it is a red color fluid that contains RBCs. RBCs consist of a red color pigment known as hemoglobin. Now lymph, it contains plasma and lesser number of WBCs and platelet, and blood, it contains plasma, RBCs, WBCs as well as platelets. Now let's talk about lymph. It helps in body defense and is a part of the immune system. Blood, it is associated with the circulation of oxygen as well as the carbon dioxide. Lymph, its plasma lack proteins. Now let's talk about the blood. Its plasma has proteins, calcium, and phosphorus as well. Lymph transport nutrients from the tissue cells to the blood through the lymphatic vessels. Now blood transport nutrients and oxygen from one organ to the another. Now lymph, the flow of lymph is slow, and blood, the flow of blood in the blood vessel is fast. Okay, next question, what is meant by double circulation and what is its significance? So let's have a look at the answer. Okay, so double circulation means that blood passes through the heart twice during one complete cycle. Now, this type of circulation is found in amphibians, reptile, birds, and mammals. However, the way it's structured and how efficiently it worked can vary between the different groups. For example, in mammals and birds, the system is more advanced as well as efficient. In birds and mammals, double circulation is especially prominent because their hearts are completely divided into four chambers: right atrium, left ventricle, left atrium, and left ventricle. And this complete separation is like having two separate pumps in one heart, which prevents mixing of the oxygen rich and oxygen poor blood and draws for more efficient oxygen delivery. Okay, now let's look at the two main parts of double fertilization. Okay, first is the systemic circulation. This is the pathway where oxygenated blood is pumped from the heart to all the tissues of the body, delivering oxygen and nutrients. Okay, so this is known as systemic circulation from heart to rest of the body tissues. Second one is the pulmonary circulation. So here, deoxygenated blood is sent from the heart to the lungs to pick up fresh oxygen and release carbon dioxide. Let's continue with the solution. Okay, now we were talking about the systemic circulation. Now, this is the process where oxygenated blood leaves the left ventricle and enters the aorta, which is the body's main artery. Now, after leaving the aorta, the blood travels through a network of arteries, arterioles, and finally capillaries. Now, these vessels act like delivery trucks, bringing oxygen and nutrients to every tissue in your body. Now, once the blood has delivered its oxygen, it collects waste product and becomes deoxygenated. Now, this blood is then gathered by venules and veins, eventually reaching the vena cava, which returns it to the heart. Okay, now let's discuss pulmonary circulation. So here deoxygenated blood is pumped from the right ventricles into the pulmonary artery, which carries it to the lungs. So this is where the blood gets rid of carbon dioxide and picks up fresh oxygen. After the blood is oxygenated in the lungs, it returns to the heart through the pulmonary veins, entering the left atrium. So this completes the pulmonary circuit and prepares the blood for another round through the body. So we can say in the double circulation, blood alternates between passing through the lungs and the rest of the body. This ensures that oxygen-rich and oxygen-poor blood remains separate, making the system more efficient. Okay, let's continue with the explanation in the next slide. What is the significance of double circulation? First, the separation of oxygenated and deoxygenated blood allows a more efficient supply of oxygen to the body cells. This think of it as having two separate delivery routes, so oxygen always reaches its destination quickly. Second, blood circulation is circulated to the body tissues through systemic circulation and to the lungs through pulmonary circulation. So each circuit has a specific job working together to keep us healthy. Next question, write the difference between blood and lymph, open and closed system of circulation, systole and diastole and P wave and the T wave. What will be the correct answer? Let's have a look at the answer for this question. Let's talk about blood and lymph. So here is a table comparing blood and lymph. So blood is red in color and contain RBCs, while lymph is colorless and lacks RBCs. Blood has more proteins and is mainly for transport, while lymph is important for immune defense. So you can think of blood as the main delivery system and lymph as the security patrol. Okay, so here in this table you can see the first point, blood is a red colored fluid that contains RBCs. Now lymph is a colorless fluid that lacks RBCs. Option number 2, it contains plasma, RBC, WBC and platelets. It also contains some proteins as well. Lymph, it contains plasma and lesser number of RBCs and platelets, and it lacks proteins. Now, the blood transport nutrients and oxygen from one organ to another. This is the function of the blood transport of the gases. Okay, elimination of the waste product as well. Now, lymph plays a role in the defensive system of the body, and it's a part of the immune system. Next question. Okay, let's continue with the answer. Okay, so another one was open and closed system of the circulation. Now, arthropod and certain mollusks such as snails have an open circulatory system where hemolymph, okay, which is blood in these arthropods, it flows freely from the heart into body cavities or the sinuses, directly bathing organs and tissues in a low pressure, less efficient system that lacks precise blood flow regulation. In contrast, annelids like earthworm and echinoderms and all vertebrates, they use a closed circulatory system, a high pressure, more efficient system where blood is always confined to a closed network of vessels and is precisely regulated by valves to ensure rapid, directed delivery of oxygen and nutrients to the tissues. Clear? So you can see in open system of circulation, in this system, blood is pumped by the heart through large vessels into body cavities called sinuses. Closed system circulation, in this system, blood is pumped by the heart through a closed network of vessels. Now, the body tissues are in direct contact with the blood in case of open system circulation. In closed system circulation, the body tissue are not in direct contact with the blood. In open circulatory system, the blood flows at low pressure, hence it is slower and less efficient system of circulation. And in closed system of circulation, blood flows at high pressure. Okay, hence it is faster and more efficient system of circulation. In case of open system circulation, the flow of blood is not regulated through the tissues and organs, and in case of closed circulatory system, the flow of blood can be regulated by the valves. Okay, so these valves, they stop the backflow. Now, open system circulation, the system is present in arthropod and mollusk, and closed system circulation, the system is present in annelids, echinoderms, and vertebrates. Systole and diastole, let's differentiate between them. Okay, so systole is all about contraction and pushing blood out, while diastole is about relaxation and letting the heart fill up again. Now, this cycle keeps your blood moving efficiently, just like a pump that alternates between pushing and refilling. So, systole is basically, it is the contraction of the heart chambers to drive blood into the aorta and the pulmonary artery, and diastole, it is a relaxation of the heart chambers between two contractions. And during diastole, the chambers are filled with the blood. Now again, systole decreases the volume of the heart chambers and forces the blood out of them, and diastole brings the heart chambers back into original size to receive more blood. Okay, let's continue with the answer. Next one is the difference between the P wave and the T wave. Okay, so you can see The P wave is linked to the activation of the SA node and atrial contraction, while the T wave is related to ventricular relaxation. Okay, so P wave. So in an ECG electrocardiogram, the P wave indicates the activation of the SA node, and T wave, okay, the T wave represents ventricular relaxation. Now let's talk about the P wave. During this phase, the impulse of contraction is generated by the SA node, causing atrial depolarization. Let's talk about the T wave. During this phase, the ventricles relax and turn to their normal state. Okay. Now let's talk about the P wave. It is of atrial origin, and T wave, this is of ventricular origin. Next question, describe the evolutionary change in the pattern of heart among the vertebrates. Okay, so let's have a look at the answer. Now, all vertebrate possess a heart, a hollow muscular organ composed of cardiac muscle fibers. Now, the heart's main function is to pump oxygen to every part of the body. Now, a major evolutionary trend in the heart is the separation of oxygenated and the deoxygenated blood. Now, this separation allows for more efficient oxygen transport, which is especially important for animals with higher energy needs like birds and mammals. Now, in the earliest vertebrates such as fishes, the heart was very simple, almost like a hollow tube. So, this basic design was enough for their aquatic lifestyle, but it didn't allow for much separation of oxygen rich and oxygen poor blood. Over time, the simple heart evolved into the four-chambered heart found in mammals. So this advanced design allows for complete separation of oxygenated and deoxygenated blood, making the circulatory system much more efficient. Okay, let's continue with the discussion in the next slide. Okay, let's talk about the Piscean heart. Okay, means heart which is present in the fishes. Okay, so this is a starting point for understanding how the vertebrate heart has evolved. Now, fish have two chambers in their heart, one auricle and one ventricle. Now, because both the auricle and ventricles are undivided, only deoxygenated blood passes through the fish heart. So this means the heart doesn't separate oxygen rich and oxygen poor blood, unlike in more advanced animals. Now, the deoxygenated blood from the ventricle is sent directly to the gills for oxygenation. In addition to the main chambers, fish heart has extra parts called the sinus venosus and conus arteriosus. Okay, in this image you can see the Piscean heart. It has one auricle and one ventricle. Okay, and you can see the sinus venosus and coronus arteriosus also. Okay, these are the additional chambers. Okay, let's talk about the amphibian heart now. Okay, so this is more complex than fish. Now, amphibians like frogs have a heart with three chambered. Okay, what are these three chambers? Two auricles and one ventricle. So this setup allows for some separation of oxygenated and deoxygenated blood, but not complete separation, which occurs in mammals. The right and left auricles are separated by a wall, but the ventricle is undivided. So this means that while the blood enters separately, it mixes in the ventricles before being pumped out. In addition to the main chambers, amphibians have extra structures called the sinus venosus and conus arteriosus. Okay, these help manage the flow of blood into and out of the heart, acting like extra checkpoints or valves. Now, oxygenated blood from the lungs enters the left auricle, while deoxygenated blood from the body enters the right auricle at the same time. So this dual entry system is important for amphibians, but because the ventricle is undivided, some mixing of the blood still occurs. Okay. Now, both these auricles empty into the ventricle, wherein the oxygenated and deoxygenated blood gets mixed to some extent. Now, this partial mixing means the blood sent to the body isn't full of oxygen rich. Okay. Now, reptiles, let's talk about the reptile. They have incomplete four-chambered heart, except for crocodiles. Okay. These crocodiles, alligators, and gharial, they have four-chambered heart. So, they are the exception. Another feature of reptilian heart is the presence of only one accessory chamber called the sinus venosus. So this chamber acts like a waiting area for the blood where it enters the main heart chamber, helping it to regulate the blood flow. Here you can see this is the typical reptilian heart. The reptilian heart has three chambers, two atria, and one partially divided ventricle, which result in some mixing of oxygenated and the deoxygenated blood. Okay, now the main exception here is crocodile, which has four-chambered heart, similar to birds and mammals. Now, the three-chambered design in the reptiles, this represents an evolutionary step between the amphibian heart and the more efficient four-chambered heart of the mammals. Okay, clear? So this is an evolutionary step between the amphibian heart and the heart of the mammals as well as the birds. And this provides a more effective circulatory system than amphibians, while still being adapted to the lower metabolic needs of most reptiles. Okay, in reptiles, the heart still allows some mixing of oxygenated and deoxygenated blood. This means the circulatory system is not as efficient as that of birds and the mammals. Again, this is an image showing you reptilian heart. Now let's talk about avian and the mammalian heart. So these represent the most advanced stage in the vertebrate heart evolution. They have two pairs of chambers for separating oxygenated and deoxygenated blood. Now the heart in these animals is divided into four chambers. Now this complete separation is like having two separate pumps in one organ, making the circulatory system highly efficient. Okay, now the upper two chambers are called atria and the lower two are known as the ventricles. The atria act like entry halls where blood first arrives, while the ventricles are like powerful pumps that sends blood out of the lungs and the body. Now this division is key for efficient blood flow as well. Okay, now these chambers are separated by muscular wall, also known as the septum. Now, this wall is essential because it keeps oxygenated blood from mixing with the oxygen poor blood. So, here you can see this is a mammalian heart. Okay, you can say a human heart. This is a muscular organ, roughly the size of a fist, and it is located in the chest, slightly to the left. That acts as a powerful pump for the circulatory system. Now it has four chambers, two atria and two ventricles, and is divided into right and left sides to manage deoxygenated and oxygenated blood separately. The heart's main function is to circulate oxygen and nutrient-rich blood throughout the body, delivering them to tissues and organs, while also collecting waste product and sending deoxygenated blood to the lungs to be reoxygenated. Okay, clear? So in this image, you can see this is a, you can say, human heart. It has right atrium, left atrium, right ventricles, and left ventricle. And you can see the septa also here. Next question. Why do we call our heart myogenic? So let's have a look at the answer. Now, in the human heart, contraction is initiated by a special modified heart muscles known as the sinoatrial node, also known as the SA node. This SA node is located in the right atrium. Now, the SA node has the inherent power of generating a wave of contraction and controlling the heart's rate. Hence, it is also known as the natural pacemaker. Now, since the heartbeat is initiated by the SA node and the impulse of contraction originates in the heart itself, the human heart is termed as myogenic. Okay, now the hearts of vertebrates and mollusks are also myogenic. Now let's move on to the next question. The question is sinoatrial node is called the pacemaker of our heart. Why? So let's have a look at the answer. See, the sinoatrial node or SA node is a specialized bundle of neurons located in the upper part of the right atrium of the heart. Now, the cardiac impulses that start in the SA node trigger a sequence of electric events in the heart. This impulse tells the heart muscles when to contract, ensuring that blood is pumped out of the heart in a coordinated way. Finally, since the SA node both initiates and maintains the regular rhythm of the heart, it is known as the natural pacemaker of the body. Now, without this rhythmic signal, our heartbeat would be irregular or might even stop. So, the SA node is truly essential for life. Okay, next question is, what is the significance of atrioventricular node and atrioventricular bundle in the functioning of the heart? So let's discuss the answer. Now you can see the AV node is located in the right atrium. AV node is clear, atrioventricular node. This is located in the right atrium near the base of the wall that separates the two upper chambers. Now, this strategic position allows it to receive the electrical impulses from the atria and prepare to pass it to the ventricles. Okay, now the AV node give rise to the bundle of His. So, this bundle acts like a highway conducting the electrical impulses from the atria to the ventricles. As the bundle of His travels down the heart, it splits into two branches, one for each ventricle. Now, this division allows both the right and the left ventricles to receive the signal almost at the same time, ensuring that both sides of the heart contract together for efficient blood pumping. Okay, let's continue with the answer. Okay, let's focus on the Purkinje fibers now. So these are the end branches of the heart's conducting system, forming a network that penetrates deep into the myocardium. Okay, clear? So now we see how the wave of excitation from the SA node leads to contraction of the atria, which then stimulates the AV node. So this triggers the contraction of the ventricles through the bundle of His and Purkinje fibers. Now you can say the atrioventricular node and the atrioventricular bundle are essential for the contraction of the ventricles. Without them, the heart would not be able to pump blood efficiently to the lungs and the rest of the body. Let's discuss next question. Define a cardiac cycle and the cardiac output. What will be the correct answer here? Okay, let's discuss the answer. The cardiac cycle is the complete cycle of event in the heart from the beginning of one heartbeat to the beginning of the next. Okay. Now this cardiac cycle, this includes three main phases: atrial systole, ventricular systole, and complete cardiac diastole. Okay, let's talk about cardiac output now. So cardiac output is the amount of blood pumped out by the ventricles in a minute. Okay, next question, explain heart sounds. So let's have a look at the answer. See, heart sounds are the noises made when the heart walls open and close as blood moves through the heart. In a healthy individual, there are two normal heart sounds called lub and dub. The first heart sound called lub, this is the sound you hear at the start of each heartbeat. It is usually a bit louder and longer than the second sound. Now, lub sound happens when the tricuspid and bicuspid walls they close at the beginning of the systole. Okay, which is when the heart contracts to push the blood off. Now, second heart sound dub is produced when the semilunar walls closed at the start of the diastole, which is when the heart relaxes and fills with blood again. Okay, so this is the second heart sound dub. When it is being produced, when the semilunar walls close at the start of the diastole. Okay. Now, listening to these heart sound gives doctor important information about how well the heart is working. If the sound is abnormal, it can be a sign of valve problem or other heart issues. Next question, draw standard ECG and explain the different segments in it. Okay, now let's discuss the answer. So see, what is electrocardiogram? So ECG, it's an graphical representation of the cardiac cycle produced by a machine called an electrocardiograph. Okay, or electrograph. Now here you can see the diagrammatical representation of a standard ECG. Okay, now typical human ECG have 5 waves P, Q, R, S and T. Now, each wave represents a specific event in the heart's electrical cycle. Okay, the P, R and T waves are above the baseline and are called positive waves. This means the electrical activity is moving in a certain direction, which is important for understanding how the heart contracts and relax. So here this is an image showing you ECG. Okay, you can notice the shape and the position of the waves P, Q, R, S and T. Okay, let's continue with the discussion of this question. Q and S waves are negative waves, meaning they fall before the baseline of the ECG. This is important because it helps us identify the different phases of the heartbeat. Okay, now P waves, they come from the atria, while the Q, R, S and T waves are all generated by the ventricles. So this distribution helps us understand which part of the heart is active during each wave. The P wave shows atrial depolarization, which is when the atria are getting ready to contract. Now the SA node or sinoatrial node starts this process. The PQ wave, which follows, represents the actual contraction of the atria pushing blood into the ventricles. Now the QR wave comes just before the ventricles contract. It represents the electrical impulse moving from the AV node to the ventricular walls, leading to ventricular depolarization. Okay, so this is a key step that triggers the strong pumping action of the ventricles. Okay, this is an image showing you a typical ECG. Okay, now let's focus on the RS wave. So RS waves is the part of the QRS complex and specially represent the contraction of the ventricles. And this lasts for about 0.3 seconds. So this is the main forceful phase of the heartbeat, pushing blood out of the body and the lungs. Next is the ST waves, or more accurately the ST segment. This represents the relaxation of the ventricles and last about zero point 0.4 seconds. And during this time, ventricles are returning to their normal state, preparing for the next heartbeat. And this phase is crucial for heart to rest and refill with the blood. Now, the T wave follows the ST segment and also represents ventricular relaxation. And this wave shows the final recovery phase of the ventricles before the next cycle begins. A normal T wave is a sign that the heart is recovering properly after each beat. Okay, again, this is an image showing you ECG. Okay, so with this, we end this session. I hope the session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.